Gap junctions have been suggested as drug targets. Iodide-YFP-GJIC assay is compatible with high-throughput screening to discover gap junction modulators. It can be used to test the effects of a large number of compounds on gap junction activities in a relatively short period. Iodide-YFP-GJIC assay is simple, robust, rapid, and inexpensive. This assay requires only acceptor and donor cells and 2 balanced salt solutions. No additional reagents such as lucifer yellow and calcine AM are needed. Also, it measures total gap junction activities of the cells in a single well, which results in low between-well variability.
To begin this procedure, grow LN215 cells to 80% confluency in supplemented DMEM as outlined in the text protocol. 1 day before transduction, wash the cells twice with 10 milliliters of PBS. Add 2 milliliters of 0.25% trypsin EDTA to the cells and incubate at 37 degrees Celsius for 3 minutes. Using a 10 milliliter serologic pipette, resuspend the cells in 5 milliliters of culture medium and adjust the cell density to 50,000 cells per milliliter. Add 400 microliters of media, which contains 20,000 cells, to each well of a 24-well culture plate. Incubate at 37 degrees Celsius for 24 hours. The next day, transduce 2 wells by